question, Aisha. Aisha, you want me to repeat the question? Yeah. In the starting of the session, we said the cell wall of plant cell will help to resist the environmental conditions. Okay. And the plants to maintain the balance in plant cell. So when you take animal cell, how that balancing with various conditions inside animal cell will be done by plasma membrane. Turbo pressure happens inside animal cell. That's my question. Not turbo pressure. No, not exactly turbo pressure. Yeah, there's no cell wall. No, that's what. There's no cell wall. Then how the plasma membrane will help the animal cell to get into an equilibrium? That's my question. Osmosis. Good attempt, Ronak. Yes. Other than that, any other answers other than that? Osmosis is halfway of the answer. Ascent of sap. Veda. Nice to hear. Good terminology. Good, very good, Veda. You know ascent of sap.
Working again.
Arya? Pardon? I didn't get.
Working again.
It's glitching again. Hypotonic solution. Yes, yes. Good, good. Then no problem. If you didn't get any idea of these things, I'll explain now. Okay. So here, assume that this is the cell, inside of a cell. Only a part was shown so that the plasma membrane function can be discussed in a proper way. So this is the plasma membrane. This dark blue one, the whole part is inside of the cell. Okay, cytosol is nothing but the cytoplasm of the cell. And this is all the extracellular fluid. Extracellular means what? Outside the cell. Okay, outside the cell. This light blue shady part is outside the cell. And this is inside the cell. In between you have the plasma membrane. Okay. Now we'll see how the plasma membrane will maintain balance inside an animal cell. Because we won't have cell wall in animal cell like the plant cell. Now when you take various terminology over here, phagocytosis, pinocytosis, receptor-mediated endocytosis. All these words are related to the intake of substances from outside the cell into the cell by plasma membrane. Okay? Phagocytosis, pinocytosis, endocytosis. And all the words, if you observe, cytosis is the common part of the word, isn't it? Cytosis. So cytosis is nothing but taking in the outside material by the plasma membrane. How this happens? Now let's see. When you take the word phagocytosis, phago means solid particles. The solid particles will be taken in by the cell by a process called phagocytosis. Particles include solute molecules like glucose or proteins or lipids, which you will intake as nutrients in food, will be taken inside the cell by the plasma membrane. Okay. The ingested material here you can see the ingested material word. Ingested means what? What are the materials, nutrients you take along with your food? They'll be taken in by a process called phagocytosis. Okay, phago is nothing but solid particles. Cytosis, taking in, intake. Okay, how that happens? So assume that you chewed some food, a bread or some biscuit, something like that. So that is the material. Here we are showing as extracellular material. Okay, it may be a protein or it may be a carbohydrate. Now once you are chewing in, the cells has to take in what the material you chewed. Suppose assume that this is the plasma membrane of the digestive system stomach part. What happens is the plasma membrane will develop structures like pseudopodia. I hope you remember this word with amoeba, isn't it? So the pseudopodia-like structures will be formed by the plasma membrane so that the taken in materials will be pushed into the cell of stomach or intestine, you can say. So here the ingested particles like the digestive material or foreign substances also like the bacteria and all, the unhygienic material also will get ingested. So taking in the solid particles is the phagocytosis process. Now when you take pinocytosis, pino is nothing but taking in fluid material. Okay, so cytosis is always taken in the material. If some fluid material was taken, how the cell membrane will take it? It will not form any pseudopodia. It will just make some vesicles. So this is the term called vesicle. Vesicle is nothing but bubbling off. A kind of bubble will be formed by the plasma membrane. It will take in the fluid material and it will push into the cytoplasm of the cell. So this is called pinocytosis. Pinocytosis is the word related to intake of fluid material by the plasma membrane from outside into the cell. Okay. Now, when you take endocytosis, endo, here endo means internalizing. Here comes the word receptor. We remember the word receptor? Receptors are the proteins which you will find on the surface of the plasma membrane. We discussed regarding the blood grouping, isn't it? So here they used the word clathrin. Clathrin. Okay, clathrin is nothing but a protein. So protein receptor, see, you can see the structure here, protein receptor. Yeah. So here the receptors will accept some molecules and they will push the outside extracellular molecules through the receptors into the cell in the formation of vesicles again. Vesicles. Vesicle is nothing but a bubbled off part of plasma membrane. Okay, like this. So here we discussed 3 processes by which cell will take in food material either in the form of solid particles or in the form of liquid matter or through receptors. Each process is termed with different words. Phagocytosis, intake of solid material. Pinocytosis, intake of liquid material. Endocytosis, internalized by receptor mediation of intake material. So these are the 3 terms we'll find across with plasma membrane of animal cell. Am I clear with these 3 concepts? Are there any doubts?
Yes, ma'am.
Okay, if you are not having any doubt, let us get into the next point of what if some different molecules tries to enter into the cell. Okay, for that purpose we have to discuss about cell structure in detail. Okay, So exactly we are now getting into the cell structure. So far we discussed only the outer membranes of the cell. In case of plant, we discussed about the cell wall, structure and function. In case of animal, we discussed about the plasma membrane, structure and function. Now this is how exactly students are getting the full screen. Is the slide visible to everyone? Here if you see, if you see, this is the whole cell. Here they have taken example of which cell? Is it a plant cell or animal cell? Yeah.
Animal cell.
Animal cell. It's an animal cell. How you can conclude that it is an animal cell?
Ma'am, because it has no cell wall. Ma'am,
No cell wall. Okay. Other than that,
it doesn't have a cell wall.
Other than that, here cell wall is not labeled, so you are saying it's a cell wall. But just seeing the picture itself, you can conclude whether it is a plant cell or animal cell. I gave a very important discrimination. Yes, Feather, very good. Yes,
There isn't any chloroplast.
No, not that point area.
And the nucleus is very big.
Yes.
Or like
yes,
the cell is round.
Yes, Feather, good. Nice. Excellent, Aisha. Yes, Shamik. Yes, section is actually smaller. Okay, so if you see a big nucleus, we can conclude it as an animal cell. Say suppose if you have the physical sessions of class, if you come across with the microscope uses, just by seeing a slide under microscope, you can immediately conclude with big vacuole plant cell, with big nucleus animal cell. Yes. Okay. Now here, this is an animal cell with plasma membrane. The plasma membrane structure they showed as lipid bilayer. Okay. Then after that you are having various parts of the cell. So we'll discuss each of this part in detail in the coming sessions. Okay. Now just concentrate only the liquid material which is there inside the cell. Okay. So when you take animal cell or plant cell, you will have a liquid matter filled inside the cell which protects all the different parts of the cell. So that is called cytoplasm. It is also given as cytosol in some textbooks also. Both the words are same. Cytosol, cytoplasm. Okay. Can you see the slide? Okay. Here
Yes,
they have taken a cell. Okay. Now in the cell, we are having different parts of the cell. All the cells will be freely floating in a liquid material called cytoplasm.